All right, so today I'm going to talk about a project we recently published at the start of the year where we used a novel cell-free nucleic acid assay to investigate the androgen receptor in advanced prostate cancer patients. So Felix gave a great introduction into advanced prostate cancer and prostate cancer progression. I'm not going to bang on about it too much here, but I just want to, reinforce how across the entire disease spectrum of prostate cancer, even in the castrate-resistant state, continued androgen receptor pathway signaling is key to maintenance of tumour growth and tumour survival. So in metastatic castration-resistant prostate cancer, or mCRPC, there are a number of therapies available to target the continued AR signaling that occurs. So these are the androgen receptor pathway inhibitors, or ARPIs, is what I refer to them as. And in addition, there are also taxane chemotherapeutics available as part of the therapeutic regimen available to mCRPC patients. Unfortunately, however, despite these agents significantly improving quality and quantity of life in many patients since the introduction around a decade ago, mCRPC is considered to be the lethal phenotype of prostate cancer. And the development of resistance to these agents is inevitable. And in addition, there's quite a significant proportion of men who present at baseline with primary resistance. So even though these therapies, as I mentioned, have been around for quite a while now, we still have no robust leads on predictive or prognostic biomarkers. So this remains really an urgent need so we can help guide and improve care for these men. So again, one of the main drivers of therapeutic resistance in mCRPC is the reactivation of AR signaling once again. So this can be achieved through a number of mechanisms, and the two I'm going to talk about today are AR copy number gain or amplification and the expression of AR splicing variants. So firstly, AR gain has been observed in up to 60% of patients. It's quite a common aberration and has been shown now quite extensively in both solid and liquid biopsy studies to be associated with primary resistance to AR-targeted therapy. However, what we also know now is that this biomarker alone does not explain the entire etiology of tumour response and resistance. So not all patients with AR gains are resistant to therapy, and not all patients with poor outcomes have altered AR. All right. And additionally, AR splicing variants have been associated with therapeutic resistance as well, and are present in up to 30% of mCRPC cohorts. So there are around 22 reported alternative splicing variants of the androgen receptor. However, the most common are ARV7 and ARV9. So these variants or isoforms are both constitutively active. They're capable of initiating AR signaling by their intact N-terminal domain, even though they lack the canonical ligand binding domain of the canonical AR protein, which is the target of AR pathway inhibitor therapies. So conceptually, I guess it's easy to see why these constitutively active isoforms can be associated with resistance to AR therapy. And of course, there have been a number of studies, primarily now using circulating tumour cells, that have investigated the impact of ARV expression in advanced prostate cancer, but the significance of them as a biomarker is still fairly unclear. So some studies suggested that ARV expression is predictive of response, whilst others have found no association between ARV expression and clinical outcomes. So I think it's becoming clear to us now that one marker or one type of aberration is just not going to cut it as a robust biomarker in mCRPC. So we need to be at the very least performing both genomic and transcriptomic investigations simultaneously, so we can get a better idea, of aberrant pathway activation, particularly in the AR. So based on all of this, we hypothesized maybe that dual assessment of both AR DNA alterations and RNA splicing events would better identify patients with an aggressive clinical phenotype and with poor outcomes on these systemic therapies. So in order to explore this, we needed to be able to perform dual DNA and RNA analysis of the AR. So we utilised a novel first-in-class, what we call dual analyte assay, to allow us to interrogate both plasma, cell-free DNA and cell-free RNA in our patients. So this assay comes from our collaborators, Fredison, who are based out of San Francisco. They have a pan-cancer multi-gene Nextgen sequencing assay that utilises the molecular barcoding or unique molecular identifier chemistry, so they can tag unique fragments that facilitates the identification of low-frequency somatic variants present in plasma nucleic acids. So this assay approach requires the collection of a single tube of blood, so 10 mils around about in an EDTA tube is usually sufficient. They require around 40 nanograms of matched germline DNA, which is fragmented, as well as a minimum input amount of 5 nanograms of cell-free DNA and 1 nanogram of cell-free RNA. So from this material, they generate enriched targeted libraries using hybridization capture chemistry, and that obviously includes targeting the AR. So following the hybridization capture, they perform massively parallel sequencing on an Illumina platform, and the aim is to really generate a high unique depth of coverage across the targeted region. So that's obviously required for the high sensitivity that's important for a liquid biopsy approach where the tumour nucleic acids can be highly diluted in normal cell-free DNA. So the sequencing data enters an optimized bioinformatics algorithm. So low quality reads firstly are discarded. The remaining reads are clustered based on this molecular barcoding technique in order to generate error-corrected consensus reads, which hopefully removes the majority of sequencing and PCR errors. So what you end up with is somatic variants. So these are point mutations, insertions and deletions, copy number alterations, and splicing variants being identified. And the algorithm also removes known benign variants by searching publicly available databases. So in addition, what becomes important here is the matched germline DNA samples that's used to remove low-frequency clonal hematopoiesis variants that can confound somatic variant analysis. So we performed some validation studies for this assay, and I've listed some of the performance characteristics here that we published recently, along with data from a prospective cohort of MCRPC patients that I'm going to talk about in the next few slides. But for cell-free DNA, the limit of detection was 0.25% or down to 0.1% for hotspot alterations due to greater depth at those regions. For these low-frequency variants, you can see we have fairly or quite a high sensitivity and positive predictive value. And additionally, using a validation cohort, there was a 100% concordance rate between the NGS assay approach and low-pass whole genome sequencing for identifying AR copy number gains. For the cell-free RNA portion, the limit of detection for AR variants was at least 2 alternately spliced molecules in each input, so per sample. And we also performed validation studies that showed a 100% concordance rate with digital droplet PCR assay if there are at least 10 alternately spliced molecules in an input. So basically we applied this liquid biopsy assay on 67 men with MCRPC who we recruited across both Monash Health in Victoria and Chris O'Brien Lifehouse in Sydney. So we collected whole blood from these patients at baseline, so before they went on to receive a standard of care therapy such as an AR targeted therapy, enzalutamide abiraterone or a taxane chemotherapeutic. And the cohort was, as you can see here, a mix of treatment-naive patients and patients who had prior treatment exposure, so who had previously received 1 or more lines of therapy for MCRPC. So from the blood, we immediately separated it via high-speed centrifugation twice, and then we immediately froze the plasma and cellular portions at -80, and they were only thawed once for batch processing at protosome. So these samples underwent the nucleic acid sequencing using the assay I just described, and we collected targeted sequencing data. So not only did we receive AR mutational data or targeted sequencing results, but also the calculated ctDNA fraction. So the median here in this cohort was 24%. I've included this figure here at the bottom that shows the AR aberration landscape for all of our patients in the cohort separated by treatment type received. So each column is a single patient and the orange tiles are a positive call for a particular alteration type. So 54% of the cohort had at least one type of AR aberration, so you can see it's quite a pervasive or it's quite commonly altered in advanced prostate cancer. Importantly, patients with an identified androgen receptor gain did not have a significantly higher ctDNA fraction than those that were AR neutral. So this is important, I guess, data to show as it shows or signifies that the identification of copy number alterations was not solely dependent upon patients having a higher tumour burden. And lastly, we saw AR point mutations identified at a vast range of allelic frequencies in this cohort, so from 0.13 all the way up to 49% with a median of 2.1. So I'm not going to show this, all of the data in the interest of time, but in line with previous research, we saw that patients with an AR copy number gain at baseline were less likely to respond to therapy and had worse prognosis. Additionally, we also saw alone that AR point mutations and splice variant expression were not associated with outcomes. So again, this started to align with previous reports looking at single mutation types. However, when we combined the genomic and transcriptomic data and compared patients with any of the 3 types of aberrations versus those that were AR wild type, we saw that patients with any AR aberration experienced significantly worse outcomes on systemic therapy. So these 4 outcomes included short PSA progression-free survival, clinical or radiographic progression-free survival, and also significantly shorter overall survival. So you can see that here on these KM curves where AR aberrancy is indicated in the green line. And also important is that these results remain significant again after we adjusted for ctDNA fraction within a regression model. So this data I think starts to indicate that combining DNA and RNA data gives a better prediction of outcomes on systemic therapy rather than just looking maybe at AR gain or ARVs or one type of alteration alone. Additionally, and more excitingly, we also observed in novel subgroups that had exceedingly poor outcomes on systemic therapy. So these patients had concurrent AR gain and they had expression of one of the constitutively active splice variants. So they're indicated here again in green, and they made up around 15% of the cohort. So compared to patients without what we call this dual aberrant signature, the subgroup the patients had significantly shorter median time on therapy before they experienced progression. And there was also a trend towards shorter overall survival. So I'm not sure we think maybe this didn't quite reach significance due to insufficient follow-up time, or we did have quite, as you can probably see, a small cohort size. But for example, here we have a median clinical progression-free survival time for these dual patients almost 3 times shorter than the rest of the cohort. So that was quite a striking result to us. So this association was observed regardless of the type of therapy received. Remember, I said treatment was either with an ARPI or with taxane chemotherapy. So the hazard ratios I've listed here for PSA progression-free survival for both therapy types, you can see, are well above 1, which means that dual AR aberrant patients at baseline did much more poorly on both ARPI targeted therapy and chemotherapy. This is a novel observation and suggests maybe that we think that a heavily altered AR locus may be a treatment agnostic biomarker and identifies patients who are going to have an extremely poor prognosis regardless of their treatment choice. So this aligns, I guess, with what we see in the clinic, where there's a proportion of men who never seem to have any appreciable response to the currently available therapy choices. So if we can use biomarkers to identify this cohort at baseline, we can begin to explore, I guess, the wider molecular landscape of these patients in order to better understand their disease etiology and maybe potentially identify other treatment courses or trials that they may respond better to, but at the very least would provide, I guess, a rationale for more aggressive therapy and monitoring in the clinic. So since this assay is pan-cancer assay, we've got a lot of genes, we've got a lot of mutational data on a number of other targets. We've started the process of looking through this dual aberrant cohort to see whether they share any other genomic similarities. But yeah, so as I mentioned earlier, this data was published at the beginning of the year in European Urology. And as part of this project, we're able to show for the first time or validate and show the success of a dual analyte next-gen sequencing assay. So we can assess both plasma cell-free DNA and cell-free RNA from a single tube of blood from our patients. So this has allowed us to identify potentially a prognostic or treatment agnostic multi-analyte signature, and it's also allowed us to demonstrate the importance of not only looking at genomic or transcriptomic events, but doing a simultaneous investigation to get a better picture of what's going inside of patient's tumour. And I'd also like to point out the study focused on coding regions only, and we saw from Felix's talk a few minutes ago that non-coding regions are the regions pertaining to androgen receptor signaling pathway may be as important for determining AR activity. So the hope in future is that we can keep building upon the amount of information we can collect on the AR to improve biomarker selection or identification. And the last point I wanted to make was that we not only saw this link between dual AR aberrancy and poor outcomes in our own cohort, but we confirmed them in this independent validation cohort of 40 patients that were recruited by a collaborator at the Mayo Clinic in the US, which is also formed part of the manuscript. So I'm going to leave it there and just thank everyone who was included in this study, including the patients and families. Thank you.